Okay. Hello, everyone. Good morning or good afternoon, depending on where you are. Welcome to today's Agri seminar. A friendly reminder that please mute yourself and type your questions or comments in the chat box during the presentation. And we will have a Q&A session afterwards. Our today's speaker is Dr. Casey Youngflesh. Casey is a quantitative ecologist and postdoc with Morgan Tingley in the Department of Ecology and Evolutionary Biology at UCLA. His research seeks to understand how ecological systems function, how they are responding to rapid global change, and what this might tell us about how to best conserve these systems. Casey has a particular interest in applying quantitative tools to large-scale data derived from a variety of sources, including citizen science projects, satellite-based sensors, remote camera networks, and field-based efforts. His research efforts have taken him across the world, from Antarctica to the Galapagos Islands, though these days he can mostly be found at his computer trying to make sense of his data. Okay, Casey, please take away.
All right, great, thanks. Let's try to set the screen share up. Is that all good? Can you see that all right?
Yes.
Perfect. Well, thanks for the introduction, Wen. And thanks to everyone for being here. I'm very happy to talk with all of you over Zoom, unfortunately, but excited to tell you a bit about my work. So as Wen mentioned, I typically refer to myself as a quantitative ecologist. What I broadly seek to do is understand how and why natural systems vary over space and time, how they're responding to global change, and what this might tell us about conserving and managing these systems, so applying these things to sort of preserve the planet. So quantifying the dynamics of these ecological systems is really quite a challenging task, of course. So ecosystems are inherently complex. Nonlinear dynamics are the norm, and responses that we see are often very idiosyncratic, so showing one pattern in some cases and a completely different pattern in others. So much of what we know about natural systems is somewhat limited in scope. They're limited by the number of populations, limited geographic or temporal scope. But then we're really forced to generalize our findings widely in this effort to understand this natural world that we live in. So I would argue that if we are to understand these processes in a holistic manner, that we need to characterize ecological interactions across the entirety of the ecological parameter space. So that is, we need to explore dynamics at large spatial, temporal, and taxonomic scales. Now, I'm sure all of you are frankly fed up with people telling you about how we're in the age of big data, but we're in the age of big data. Even ecology is in the age of big data. So there are a number of new and forthcoming data streams that are just now coming online that are relevant to the field of ecology. So for instance, community source data from large-scale projects such as eBird, which I'll talk about today, data from newly developed satellite sensors, incredibly high spatial temporal resolution, things like remote camera networks. These are just a few examples of things that are really most relevant to my own work. And now we have the cyber infrastructure, the quantitative tools to better utilize long-standing existing sources of data in novel ways as well. So using these tools can develop these analytical frameworks to effectively scale up our research efforts in ways that weren't previously possible, really to answer both the fundamental and again, the applied ecological questions. So as NSF has eloquently put it in their recent list of 10 big ideas, we have this opportunity to harness the data revolution. So with my work, I seek to do just that, to take advantage of these resources to understand how and why we observe the ecological patterns that we do. So my talk today is going to focus on phenology, so that is the timing of seasonal events. And I'll place a particular focus on how we might think about these processes in the context of global change and what responses to these environmental changes might tell us about how ecosystems function. So while birds will really be the focus of these case studies that I'll run through, I'm really interested in what this work can tell us about ecological systems more generally. My work really explores a larger set of questions using approaches similar to the ones that I'll talk about today. So phenology, again, the timing of seasonal events really plays a critical role in ecosystem functionality. So organisms need to time important life history events to coincide with events at other trophic levels. So for instance, birds might need to time their breeding with insect emergence. Raising chicks is an energy-intensive process that requires increased food resources. And the availability of those resources varies over the course of the year. So there are going to be fewer resources in winter, generally more resources during the spring and summer. And then those insects actually need to time their emergence with the availability of their own food resource, plants. So in this case, I'm using this bird, insect, plant system as an example, but really this is applicable to any system in which resources are seasonally pulsed, so these seasonal environments. So in response to recent climate change, we're now seeing a shift in when these seasonal events are occurring. So in aggregate, ecological events associated with spring are generally occurring earlier in the year than they did several decades ago. And so there's this concern now that the times of peak resource requirements are no longer matching up with periods of peak resource availability. So that is, these two windows may be advancing at different rates over time, leading to this sort of temporal decoupling. So here in this figure, the blue ribbon represents peak resource availability. The red ribbon represents peak resource requirements. And while these two windows may have at one point overlapped, over time, this may be increasingly untrue, with the potential for demographic consequences of these temporal asynchronies. So for instance, depressed breeding success. So in light of these links between phenology and demographics, I think it's really critical that we develop an understanding of how organisms are responding to phenological dynamics at these lower trophic levels, the base of the food web, so to speak. So while it's been shown that the timing of many seasonal events is changing, I think we need to move to understand how and why responses to these changes vary across species, across space, and ultimately what that might tell us about the potential vulnerability of different taxa or systems. So this is just here to give you a bit of an idea about what the annual life cycle of one migratory North American bird species might look like, and really the important role that phenology plays in that. So many species that breed in North America overwinter in Central and South America. So this is the American redstart. It overwinters down in this region, Latin America, in blue, migrates northward during the spring migration. We have the time of year around the outside here. Breeds during the summer months across North America, and then migrates back south during the fall. So these birds, of course, need to migrate at the correct time of year. Birds are doing this sort of optimization where they don't want to arrive too early and experience winter conditions, cold temperatures, less food, but they also don't want to arrive too late. It's not enough time to raise chicks before they need to migrate southward again for the winter. So phenology, the timing of all of these annual events, migration, breeding, is quite important for these birds. So I was interested in understanding how the phenology of birds, specifically in North America, is responding to environmental change. However, what we might consider traditional data resources don't have the spatial taxonomic breadth to really facilitate this work. Luckily, there are resources such as eBird, which we can use to leverage large-scale information being collected by bird enthusiasts, such as these three characters here. So if you're not familiar with eBird, it's an online platform where individuals can upload sightings of any birds that they may have seen. This is community source data collection, sometimes referred to as citizen science. So for a given arbitrary area, such as this red cell, in a given year, there will be a number of eBird checklists collected by birders throughout the year. So in some checklists, a species, say red-eyed vireo here, might be recorded. It's a record of presence. In other checklists, that species might not be recorded. That's a record of absence. Then using these presence-absence data, I modeled how the probability that a given species occurs in a checklist changes as a function of day of year. So in this plot, circles at the top represent checklists where red-eyed vireo was recorded for this particular cell year. It's a success in a Bernoulli trial. Circles at the bottom of the plot represent checklists where red-eyed vireo was not recorded. In black, we have the model fit for a logistic GAM. It's a generalized additive model. I'm using this to fit a smooth function of its probability of occurrence over the course of the year. We also have effort hours in here as a covariate. That's that EH. So the year begins with a very low probability that this particular species occurs in a checklist. And then this rapidly increases during the spring, really what we'd expect here. We then defined the point at which this curve is half of its maximum value. We call this the half max. So this is shown in the solid blue vertical line. The uncertainty there is the dashed blue vertical lines. And this half max is our metric of arrival phenology. It's really when migration really ramps up in the spring. So I ran these GAMs for each cell over our study area. So it's everything east of 100 degrees west longitude here, eastern US and Canada, for each year, for each of 56 different species, which amounts to over 7 million bird observations from eBird. So here I'm showing these GAM-derived arrival estimates for red-eyed vireo for a single year. It's 2006. At the left, this is the same plot that I showed on the previous slide, fitting that GAM to estimate this half max. So in the map at the top right, the colored cells represent regions where we were able to reliably fit these GAMs for this particular year. So really relatively sparse data year in 2006. The color corresponds to the date of arrival for this species for a given cell for a given year. So blue colors, later arrival dates, red colors, earlier arrival dates. So you have this gradient as you move northward as we expect. Okay, so now we know that these cells next to one another should have somewhat similar arrival dates. So in other words, we know that there's substantial spatial autocorrelation in these data. To take advantage of this spatial structure, I use a spatial autoregressive model to help estimate these arrival dates. So I use these half max estimates and the associated uncertainty there from these GAMs as inputs for this IAR model. It's an intrinsic autoregressive model. That's the spatial model. So these models are all fit in a Bayesian framework, allowing us to propagate our uncertainty through the entire analysis pipeline here. So this IAR model essentially adjusts the arrival estimates for each cell based on the values of neighboring cells. And the degree to which dates are changed depends on the values of these neighboring cells, their associated uncertainty, and the estimated degree of spatial autocorrelation. So the results of that model are shown here in the bottom right. You can also see that those gaps there where we have missing information, that's actually filled in by the model using that spatial information there. Although these interpolated estimates, those actually weren't used for any downstream analysis. So using this approach, what I've done here is basically distill this immense amount of data that was crowdsourced from bird enthusiasts into this phenological data product. So each of these maps is from a different species for an arbitrary year, where the color again represents the arrival date. And I think this is really worth noting that all of this really wasn't even possible, wasn't computationally tractable even a short time ago. So even on high performance computing resources, the computational burden was pretty substantial. So a few years of compute time. So using these estimates, I wanted to quantify how sensitive bird phenology is to the arrival of spring and how and why that might vary across space and among species. So one of the most prominent metrics of spring phenology is when plants start to green up in the spring. So this is typically referred to as green up. I wanted to compare interannual fluctuations in bird arrival to these interannual fluctuations in green up. So in the plot right here, fluctuations in bird arrival are shown in blue. Fluctuations in green up are shown in red. And those green up estimates are derived from satellite-based sensors. So these are the results for a single hexagonal cell for a single species. So showing just tree swallow here. So using a hierarchical spatially varying coefficients approach, I modeled the relationship between these two metrics, between bird arrival and green up. I wanted to know how arrival changes as a function of these fluctuations in green up. In other words, I was interested in estimating what I refer to here as sensitivity. That is, for every 1 day change in green up, how much does arrival change for a given species cell? How sensitive are birds? So a value of 1 here would represent a perfect synchrony between bird arrival and green up. So in this plot here, the dark green hues represent a higher sensitivity. The yellow hues represent lower sensitivity for this species. So this is a map of sensitivity and not arrival here. So using data through time to derive these estimates. What we find is that most species are actually more sensitive to green up in the more northerly portions of their ranges. So there's this spatial variation that's apparent even within species. We also find substantial differences in sensitivity among species. So both in the overall degree of sensitivity as well as the spatial patterning. So for instance, prairie warbler here in the bottom right shows lower overall sensitivity to green up, as well as a much smaller gradient in that sensitivity across latitude compared to tree swallow. So again, most of the 56 species I looked at exhibit this monotonic increase in sensitivity with latitude. We have sensitivity on the y-axis here, latitude on the x-axis. Each gray line is a species with the bolded lines representing those species I showed previously, and the red line represent the cross-species average here. So again, positive slope suggests that most species have this capacity to adjust their timing as they move northward, possibly in response to environmental conditions. Although there's certainly a limit to how much birds are able to adjust as they're moving northward. So I think one of the big takeaways here is that even the most sensitive species in the most sensitive portions of their ranges are not matching changes in green up 1-to-1. So birds are not adjusting their phenology enough to match these fluctuations in green up. And because of this failure to match these fluctuations, there's concern that as green up changes over time, and it's getting earlier over time, the birds won't be able to keep pace with these changes. So there's also the question as to why we see this diversity in overall sensitivity among species. So if we consider the species mean sensitivities. So to look at that, I modeled species level sensitivity to green up as a function of species level traits. So each point here is a species with the red dots labeled for the 4 species shown previously. What we find is that species that migrate more slowly, arrive earlier, and overwinter further north tend to be those that are more sensitive to green up. So in understanding how sensitivity varies among species, we can get a better idea of which species may be better equipped to respond to future phenological change. So in this case, perhaps those species towards the top of this plot. And results suggest that these responses to changes in green up might vary quite a lot among taxa. So one thing that my co-authors and I did for this publication, which I think is quite an interesting idea, is to create an interactive web-based application to explore these results. I think when many people see these figures with 56 different species, they're left feeling like they want more information, perhaps how their favorite species are responding, or maybe how this plays into what they might be observing while out hiking or birding. So this is really a tool where users can explore results of the study and actually download these data products. So I think this has really the potential to be quite a useful tool, not only for research, for downstream applications, but also something like education. So if anyone's ever looking for example data sets to teach data manipulation skills, statistics, I think this is really a great resource. It's really variation over time, space, species, and this is all freely available. And I should note this is created with the R package Shiny, a relatively user way to create these interactive web platforms. So beyond migration, of course, there are many other important events throughout the annual cycle of a migratory bird. So thus far, I've been talking about the spring migration, northward migration there. But we're also interested in when these birds are actually breeding. Breeding is arguably the phenological metric of greater interest, at least if our hope is to understand the demographic consequences of these phenological dynamics. So I've also been using eBird to look at this, but moving beyond just the presence absence that we might get from these data and using something we call breeding codes. So using this eBird platform, bird enthusiasts might note any breeding behaviors that they observe. So there's a set of standardized codes for this. So this is the platform. If you were to go on eBird.com and register an account, make a checklist, this is what you'd see. There are a number of different behavioral codes that corresponding to varying levels of certainty regarding whether a bird is actually breeding. So let's say we observe one Carolina wren, this bird pictured here, and it was exhibiting some sort of breeding behavior. So you can click on this menu and presenting us with all these different codes, possible evidence of breeding, confirmed evidence of breeding, etc. So in addition to the certainty of breeding, each of these codes is also going to indicate a different stage of breeding. So that breeding stage on the full annual cycle there of migratory birds that I showed earlier. So we might have the egg stage, code nest with eggs, NE, indicate that that species is undergoing, is at that particular stage of breeding at that particular time. The young stage, codes like nest with young, NY, indicate that birds have young in nests at that particular time of year. Or the fledge stage. So after these birds are old enough to actually leave the nest, we say they're fledging. So recently fledged young, that would be another breeding code that a birder might enter into eBird. That's something that we'd want to use. So the approach used for estimating bird arrival that I showed you earlier, I treated each checklist in a given cell for a given year as a trial, and each record of a species as a success in that breeding trial. So in modeling breeding, instead treating each checklist with the detection of a given species as the trial, so it was considered a success previously, and each record of a particular breeding code as a success in that breeding trial. So those successes are the circles at the top of the plot. So this plot here is saying conditional on a given species being recorded in a checklist, what is the probability that a breeding code associated with each of these stages, egg, young, fledge, was recorded? So because the data on breeding are much more sparse here compared to just presence absence, the idea here is to estimate when each of these stages occurs in a given cell for a given species on average. So it's more difficult to break these things into a per year basis compared to just the migration phenology. So I fit a separate GAM here, estimated these half max values for each of these breeding stages, and aggregated the checklist across all years. So one model for egg, one model for young, one model for fledge for each of these species cells. So here are some preliminary results for a single species, eastern kingbird. Each of these points for the different breeding stages represents one GAM, the results from one of these GAMs, and error bars are one posterior standard deviation about that half max. So the average arrival dates down here in this greenish color, those are derived from the results I presented previously. These are just for the cells that the species breeds in. So if we move later in the year, after arrival, we have the egg stage that's shown in orange here. After that, we have the young stage after the eggs hatched. And then once those young leave the nest, we have the fledge stage. That's the latest point in the year. So one, I think, interesting thing that initially pops out here is that these lines don't appear to be parallel. That is, the length of these different breeding stages is not constant across latitude. So here on the x-axis, we have latitude, and we have the day of year on the y-axis. So we're getting later in the year as we move up this y-axis. So using these results from a number of different species, we can start to look at these sorts of patterns in more depth. So here are some preliminary results modeling the interval between the arrival and egg stage as a function of latitude. So this is done in a fully Bayesian framework, accounting for the uncertainty in these estimates using species cells with paired data. So only species cells where both arrival and egg stages were estimated. So here each color represents a different species, and ribbons representing the uncertainty about that. So we call what I'm referring to here as the waiting period, the interval between the arrival and the egg stage on the y-axis, and we have latitude on the x-axis here. The generally negative slopes here suggest that there is this compression of this waiting period at these more northerly latitudes. So birds breed more quickly after arriving at their breeding grounds in these more northerly areas. So really what appeared to be the case in the plot on the previous slide. So these results are really quite interesting in the context of some previous work that showed that this interval here seems to be compressing over time in some cases as well. So species can only compress this period a finite amount before something has to give. They have to arrive before they lay eggs, of course. But how much time do they need? So this suggests that populations at more northerly latitudes are possibly more at risk of these phenological changes. So this is really what we're looking into here. More study is needed, but we do see that they're at these more northerly locations, we're seeing a compression here, and there's concern that through time, there's not as much wiggle room, for instance, as we continue to see a compression of this period moving into the future. So as I noted, information on bird breeding is relatively sparse. You have these gaps over space or time or for certain species, and there's really a lot of uncertainty about some of these estimates when it comes to breeding. So what I'm now doing is using statistical data integration to combine sources, different sources of data to better estimate these parameters. So estimates when really estimate when each of these stages occurs and how this varies over space and among species. So the idea of data integration here is to incorporate information from multiple data sources that are measuring the same thing or very similar things, potentially in different ways. So this figure is a conceptual representation of one way to formally integrate multiple data sets into a single analysis. So we have data set 1 here, it's the Y1s, data set 2 here are Y2s. These are both modeled as a function of Z. So we can think of these Ys as our observations, realizations of some true latent process. But the Z is really what we're interested in. This is the latent state that we're estimating using these multiple data sources. This latent Z can then be modeled as a function of covariates, whatever it is that we might be interested in. And this would all be done within a single model. So all modeled hierarchically here. So very sort of generic framework for doing this data integration. There are, however, many other ways to integrate data from multiple sources. We can incorporate information using informed Bayesian priors. So we have this prior information that we can help estimate some of these parameters in our model. So the idea here really is to use all available information to get at these questions. So in this particular case for bird phenology, we have things like eBird data that I showed you previously, but we also have bird banding data. So birds that are banded, little metal bands, data from nest monitoring programs where you might have very intensively studied areas where people are going and checking nests on a daily basis. It might have a more limited spatial scope. Each of these different sources of data comes with different degrees of uncertainty about these breeding stages and really has their own pros and cons. And really the flexible Bayesian framework here that we're implementing this data integration framework in allows us to accommodate and to model that heterogeneity in the data, as well as the uncertainty and the uneven sampling that happens with these data. So currently working on this, building these models that leverage not only the eBird data, but information from all these other sources as well. So I've talked a lot about the phenology of these birds, how we might estimate it, how it varies among species, and how it's lagging behind phenology at other trophic levels, other levels of the food web there. But we might now ask the question, does this matter? What repercussions does this have for demographic processes? What are the ecological consequences of these asynchronies? While really a plethora of studies have highlighted the potential concern of phenological change, there's surprisingly little evidence of a link to demographics. This has been somewhat of a tricky question to answer. Should we be concerned about animal phenology not keeping pace with climate change? So this is something that I explored during my PhD work on Antarctic penguins. So using several decades of field data on Adelie penguin breeding phenology, breeding success from around the continent. So sites are shown here as the red dots around Antarctica. I was interested in quantifying the demographic consequences of breeding too early or too late relative to the spring phytoplankton bloom. So this phytoplankton bloom is a sort of green up for marine environments, and it's really an important driver of biological dynamics in polar marine systems, such as in Antarctica. So for this species, I found that breeding at the correct time is a necessary but not a sufficient condition for optimal breeding success. So that is, synchrony sets an upper limit for breeding success, but doesn't actually explain much variation in the mean. So we have asynchrony on the x-axis here. That's breeding timing relative to the phytoplankton bloom. And we have breeding productivity here on the y-axis. So the number of chicks fledged per pair. It's a colony-wide average. Each point here is a year of data from a particular site. The 0 represents the mean. The pluses and minuses are deviations from that mean. So what this red line tells us here is that there is a sweet spot for maximum breeding productivity. So this line represents the model fit for a quadratic quantile regression. So the model's being fit to the 85th quantile of these data in this case. So this suggests that phenology does play a role here. It sets this upper limit, but really there are a number of other factors that influence reproductive success. You can imagine that even when these penguins breed at the correct time, there might be things like extreme weather events, something that I've also worked on, or anomalous sea ice conditions that might impact breeding productivity. So there may be all these other things that outweigh the importance of phenology overall with regard to demographics for this penguin. And there are really a number of characteristics about the Antarctic ecosystem which might explain why phenological synchrony isn't more important here. So might these asynchronies be more important in other systems? So for instance, in the temperate environment of our North American bird example, are these temporal asynchronies important for demographic factors in this system? So to explore this, I've been using data from a large-scale bird banding project known as MAPS. It's the Monitoring Avian Productivity and Survivorship Program. So these MAPS banding stations are distributed across the North American continent. So each station is represented by a dot on this map and provide us with really this incredible data set, this constant effort bird banding data set. And by constant effort, I mean effort is standardized across sites and across time. So from these banding records, we can calculate an index of breeding productivity, as well as the timing at which young birds are leaving the nest. So the timing of that fledging event. So we have sufficient data here to estimate these parameters for more than 40 species and almost 4,000 species site year combinations spanning the continent here. So to estimate breeding productivity for a given species at a particular site in a particular year, we can model the proportion of individuals captured at these banding stations that are juveniles. So juveniles are birds that hatched that year. So here these icons represent all the individuals at a given species caught at a site in a particular year. The red icons are juveniles, black icons are adults. So just to give you a sense of what's going on here, at these banding stations, folks set up these nets that these birds fly into. The nets are made of quite fine material. Birds don't see the nets. People then go and collect the birds out of these nets, put a band on birds that don't have bands, and record information about that bird's, the age, the species, etc. So we know whether each bird that's captured is a juvenile or an adult. We then model the number of juveniles in a given year with the binomial distribution, where y is the number of juveniles captured at this banding station in a particular year for a particular species. And this n here in the binomial is the total number of captures, juveniles plus adults, and p is the probability that a given capture is a juvenile. So this probability p, which is going to vary by species, site, and year, modeled as a function of covariates of interest. So in this case, It's going to be phenological metrics. So this capture information also provides us with a way to estimate the time of year at which these birds are fledging. So for a given species year site, I use the mean date of the first capture of juvenile birds as an indicator of fledging date. And for environmental phenology, I use green up as before derived from satellite sensors. I then use a principal components analysis to decompose these two phenology metrics, so fledge date and green up date, into two orthogonal indices. So the two principal components from the PCA. So what we're left with is an early late index as well as an asynchrony index. So as the name might suggest, the early late index is a measure of the earliness or lateness of a year in general. So whether fledge and green up were both early or both late. Here the blue lines represent green up phenology, the red lines represent bird phenology, the dashed lines at top represent an early year, so that'd be a low value on this early late index. These solid lines below represent a late year, so high value for the early late index. This asynchrony index represents how in sync bird phenology and green up are. So for instance, did a given year have a particularly early green up and late fledge, or vice versa? So I was interested in how breeding productivity might be related to the effect of both the overall timing of the year, as well as the asynchrony among these metrics. So both of these were included in this binomial model. These are covariates on that parameter P that I showed earlier. So here we have the results from that model. We have estimated productivity on the y-axis, and we have this asynchrony index on the x-axis. Each line here is a separate species, so all species were modeled together here hierarchically. These are all brand new results that I'm showing you. What we can see here is that there is this optimum value for the asynchrony index where breeding productivity is highest. So this was modeled using a quadratic function here. So we have a situation where fledge date and green up date need to match up just so for this optimal value to be achieved. So if either green up or fledge is too late or too early relative to the other, breeding success is going to be depressed. We see something similar when looking at the early late index. So here breeding productivity on the y-axis, the early late index on the x-axis. Again, each line is a separate species. If the year is too early or too late, by that I mean both fledge and green up being particularly early, particularly late, breeding productivity is depressed. But perhaps early seasons tend to be a little bit better here in general than later seasons. So visualizing these two metrics together, so this is the overall cross-species effect, we see this peak in three-dimensional space. So we have productivity on the vertical axis and our two phenological indices on the horizontal plane there. The color of that curve represents the height of that surface, so the breeding productivity there. So this is really the relationship that we expect to see between phenology and productivity, sort of what we've theorized. We expect for there to be this Goldilocks area in yellow here. So of course, for the asynchrony index, we expect that breeding too early or too late relative to lower trophic level phenology might mean that birds are not matching the timing of their food resources. However, we also see that the earliness, lateness of the year matters as well. So the importance of overall timing might signal that factors like edge season weather are important. Anomalous weather events that might be more common perhaps in the very late or very early years, though I'm sure we can come up with a number of potential explanations here for this early late pattern as well. So these results are somewhat concerning, given that we know bird phenology is changing at a different rate from green up. This was true for migration phenology, as I showed you earlier, and it's also true for breeding phenology, for fledging year. And I think this is quite an important result, given that most of what we know about demographic consequences of temporal decouplings really comes from just a handful of species. Most of these are European bird species, and really just a few of them. To my knowledge, this is really by far the largest scale demonstration of the demographic impacts of phenology, whether considering the spatial or taxonomic scale here. So this might have an important role in understanding recent large-scale declines in North American birds in recent years. So the total abundance of these birds has seen a decline of about 30% over the last 50 years. So that's about 3 billion birds that we've lost across North America over that time period. So these are results from Rosenberg et al. Paper that came out several years ago now. So the role of phenology in these declines in abundance is currently unknown, but it likely plays a role at some level here, given these sorts of patterns that we're seeing. So one caveat with this work is that we're missing a trophic level here. Most of these birds rely not on plants as a food resource, but on caterpillars. It's actually this tritrophic food web, we call it, so 3 levels to it. So over the past couple of years, I've been working with folks on the insect side of things in this regard, trying to match up essentially caterpillar phenology and availability with bird phenology, which is really quite a challenging task given the massive diversity of different caterpillars in North America and the difficulty in surveying these at these large spatial temporal scales. People love birds and are willing to go collect data on birds. And while some people like caterpillars, I don't think the enthusiasm is quite there for insects as it is for birds. So that is all to say, it's certainly something that we're endeavoring to do, to link bird and caterpillar phenology. And I think we've really laid a lot of the groundwork for things to come. But we have really some more work to do to directly link these 2 trophic levels at the scales that we've been talking about thus far. So one thing that I really want to emphasize about this work is the importance of statistical tools in all of this. So these models that I showed you were all fit using a piece of software known as STAN. It's one of the latest and greatest tools, at least in my opinion, to fit Bayesian models using MCMC, so Markov Chain Monte Carlo. So the flexibility that this Bayesian approach affords allows us to model this heterogeneity in these data and to accommodate the non-stationarity that's often apparent in natural systems. So I think it's a really important tool to understand how ecological systems are changing and to forecast how these systems are going to change into the future. So I plan to run a workshop at some point this year focused on the basics of hierarchical Bayesian modeling using STAN. So material will be directed towards those with a basic understanding of linear regression, familiarity with R, but really no or limited prior knowledge with Bayesian modeling. So a bit of a plug here for that workshop. Stay tuned for details. An announcement will go out through the usual IDRI channels when that happens. So I'd like to thank IDRI for funding work, providing me with funds as an IDRI postdoc scholar. Thanks to NSF for funding the larger project that most of this work falls under. Thanks to the eBird team and the MAPS team, to the many folks that contribute observations to eBird, that run MAP spanning stations. These type of big data really wouldn't be possible without them. Big thanks to my collaborators on all these projects here, a lot of folks. And finally, thanks to all of you for listening, and I'm happy to take any questions.
Fantastic. Thanks, Keith. Very interesting work. So let's see. So I currently don't see any questions in the chat. Do anyone from the audience have any questions? You please feel free to raise your hand or just open your mic.
Casey, thank you for this presentation. I was curious if you would expect a change over time in breeding success if you had enough data to model that.
Yeah, it's a good question. If we see the link here between phenology and breeding success, we know that phenology is changing. Yeah, wouldn't we expect to see a decline potentially in breeding? Given enough data, yeah, I think we could pick up that signal, but how much data is required, I don't really know. So I think we do see that super clear curve pattern there but there's a lot of other things going on. So it's not explaining, 80% of the variation or anything like that. As with penguins, there are all of these other processes. Maybe there are more predators one year, maybe, aside from the phenology, the timing of their food resources, maybe there's just more food resources or less food resources in a year. So to answer the question, yeah, I think given enough of data, we could see that. And I think we should continue to look at that in the future. Do we have enough to see that right now? I don't think so. And that's probably because there are all of these other factors that play a role as well. I think one thing that I'm interested in is kind of thinking about, okay, how far down the line would we expect a really large signal? Is it going to be 50 years? Is it going to be 100 years? In 100 years, a bird's going to be completely sort of mismatched with their the timing of the environment. I think that's something that we could look at and sort of, think about. Even if we can't see those huge patterns now, when in time we might expect to see those. So yeah, good question. Which of course opens up a whole host of other questions like, are these birds evolving, adapting to these changes in phenology? And that's certainly an active area of research. Is there a selection for earlier or later breeding or for individuals that can better match the timing of resources? What kind of impacts are range shifts going to play in all this? So really a number of different interacting things here.
Casey, I also have a question. So as you mentioned across the talk, so all these processes seems quite sensitive to the environmental change and weather condition or near surface climate condition. I wonder, so are you aware of any effort that trying to build a simple model that can couple ecological system and the climate system? So basically fitting in some climatological variable, maybe even in remote regions. For example, in our field, when we talk about year-to-year variation, we really care a lot about the ENSO cycle that captures the sea surface temperature variation over tropical Pacific. That is important because it can have some predictive power to tell us how the precipitation or temperature would look like in some regional scale. So I wonder if we all case whether you have ever thought about that, or do you think we have enough data, we have enough bird data to build such kind of model to make predictions?
Yeah, definitely. So, I think on the plant side of things, we're much better about that. As the plant ecologists are much better about that. I think, ecology in general tends to have less, I don't know how to put that, just more stochasticity, I guess. It's harder to describe the system with a set of equations compared to more of the physical sciences. Plants are a little bit easier to do that because, we understand the physiological mechanisms a little bit better there with something like plants. So I think we're getting really good at, forecasting when plants are going to green up based on atmospheric conditions and build, take those climate forecasts, those weather forecasts, and then forecast in the future when plants are going to green up and what's going to happen with that. Above plants, it's a little bit challenging, but I think we could say, conditional on these expectations that we have for plants based on what we think what's going to happen with the climate system. What do we expect birds to do? I think we're kind of like just on the cusp of doing that. And we're not going to be able to do that as deterministically as we do with plants or of course with atmospheric processes. But yeah, I think it's certainly worthwhile. I think most of the studies that have been done with things like, ENSO, there has been a lot looked at, what kind of effect do these large scale climate indices, these fluctuations have on, things like birds. But it's all about sort of this correlative thing in the past, and we're not thinking so much into the future. I think in general, there's a big push now to be better about forecasting ecological dynamics. And part of that is building a reliable model to, represent these processes. Again, plant folks are better at that. But yeah, I think we're really moving towards that direction, but we have a long way to go, I think, to catch up with the physical sciences, certainly.
Interesting. So also when you show the correlation between the bird's arrival and the green up of the plants, did you ever check whether that's that kind of relationship sensitive to what kind of plant species you're looking at?
Yeah, I mean, it could be very species specific. So for this green up, it was just forest habitat. There's a lot of heterogeneity even within forests. So certainly if we were to look at agricultural areas or fields or something like that, we would see something a little bit different, because you actually don't see the green ups changing much in agricultural areas for, we might expect that. I think the species of tree though could matter. And I think, yeah, that's going to vary across the species range. We really just don't have high enough resolution data to do that. So this is all the green up data was derived from satellite sensors. Basically, look at that and see how the greenness literally is changing. We can now map tree species using remotely sensed data, but it's kind of a tricky thing to do, and we don't yet have the capacity to do that on this continental scale. So yeah, I think that's an important thing and an interesting thing to do in the future, but we didn't do it here, but it might be important.
Okay. What about wildfire? Like, of course, it's a, wildfire actually is it's a natural part of the nature, right? It's not something new, but I know everyone knows that in the past few years, at least for California, maybe somewhere else, experienced significant wildfires. Do you have any guess, or do you have any idea what that mean for birds migration and all these things that you talked about today?
Yeah, it's a really interesting point, certainly relevant for California. Some of the other folks in the lab I'm in do some work on fire dynamics, particularly with fire reliant species, things like woodpeckers. I think I have actually seen some work recently, I think that came out last year, talking about how these large smoke plumes did actually divert some birds. You could imagine that they're not going to be able to persist in these maybe burned out areas, depending on the species, or even sort of migrate in real thick smoke, things like that. So yeah, I think we would certainly expect that to impact bird migration. Most of the impacts of fire, we're going to see that in the Western US. This study was focused on the Eastern US for a couple different regions, but really it's a more homogeneous environment. I use the word homogeneous sort of lightly there. It's this deciduous seasonal forest that the stereotypical forest in the Eastern US that you think about. The West is much more variable. Water precipitation plays a huge role in phenology in the West, where maybe that's a little bit less so in the East. There's just more varied habitat, more topographical change, so it's a little bit more complicated in the West. And yeah, of course, fire, that extra wrinkle there. Yeah, I think the truth is we don't really know what the impact is going to be for the migratory patterns of these birds and where they decide to breed. If their breeding areas are burned out, I guess they're going to go to the closest area that they can find, a suitable area and breed. But yeah, a lot of those consequences are unknown.
Very interesting topic. And then, are there any other questions from audience? If not, I may ask one more. Okay, so maybe let me ask one more question. So you showed, I think probably close to the end of the talk, you shared a network called Maps that provide the data that you used. I wonder whether you can share any other resources like from other continents, Eurasia or Southern Hemisphere. Are you aware of any of those data? I guess some audiences, including myself, actually is interested in taking a look. So I'm not sure whether you know any of those that you can share with us.
Definitely. So the MAPS program is focused on the sort of breeding areas of North American birds, of these migratory birds. There's also a Latin American complement to MAPS called MOSI, which is also run by the Institute for Bird Populations. It's MOSI. So there are a number of stations sort of distributed around Central America, South America there, but it's not as extensive as MAPS. For Europe, there's certainly a lot of banding data there. I don't know what the sort of the overarching schemes are called. I know the UK has one. I think Europe might have a separate one. And as far as other programs in other areas, particularly Asia, Africa, I'm not actually sure. I'd be very curious whether there's any of these large scale networks out there. I think it's, again, we're really in that time of we need to start linking up these large scale resources together and pooling our resources to understand these things across these continental scales. So yeah,
Okay.
an area for future opportunity.
Yeah, thank you so much. Okay. Any other questions from the audiences?
Well, thanks so much for having me. Thanks for attending, and feel free to shoot me an email if you have any other questions.
Yeah, thanks a lot for the interesting talk. Really learned a lot. I'm going to stop the recording.